It is 7:47. We are recording, so it's about 12 minutes before the lecture starts. So feel free to skip ahead 12 minutes if you're watching video. Morning. Well, let's get to it. Just kind of get a head start on lab stuff. Turn this off. All right. Do attendance very quickly. Of course, if you're online and if I say you're not here, obviously it's because you're online. So as usual, send the words in before 9 a.m. to get your extra credit and to be counted as present. Van Gura, not here. Casebold is not here. Tristan is not here. Covington Griggs is not here. Kately is not here. Dow is not here. Hughes, not here. Wait, Hughes? No, sorry. Lebdy is not here. Carla is not here. Mankins is not here. Mathis is not here. We got Raines back there, right?
Yeah.
Raines is here. Reed is not here. Elise is not here. Thompson is here. Let's read that one. Yendui is not here. Elkins is here. This is somebody, well, I know I did. There's only 3 of you physically present. So there we go. Let's get started.
Morning. I'm here. You mark me absent.
Yeah, just as a reminder, if you're online, you just need to send the words in.
Yep.
At sometime before 9 a.m.
Yep.
Which is a good time to remind everybody too. Remember, the words now have changed it up a little bit because obviously I'm having issues with people paying any bit of attention. So from now on, the words of me asking you a question of some easy question of some information I just gave. Likewise, we'll get to the announcements later, just like I've been doing recently, because again, I'm sharing information that people just aren't getting. Anyway, this is where we left off. We were talking about enzymes. So back, since we had to stop in the middle of an enzyme conversation, I'll give you some more, a little refresher when it comes to enzymes. Enzymes are proteins. As far as we're concerned, they're always proteins. And they are molecules that help chemical reactions happen, right? So without these enzymes, all these chemical reactions that are happening in our cells wouldn't happen. And because of that reason, we just wouldn't have life. So it's important to life. Life wouldn't exist without them. At least not life as we know it. So they're very important. So how do they work? One of the things you need to know for the exam, and again, this is a refresher from what we talked about on Monday. What they do is they reduce the activation energy, right? So whether a chemical reaction is putting off energy or if it uses energy, regardless, it needs to lower that activation energy, or that energy that is required to start the chemical reaction. And that's what enzymes do, right? And then right before we left on Monday, we started talking about this stuff here, exactly like how do they lower the enzyme energy, right? So that's how do they lower the activation energy? This is the conversation that we left off with, right? We talked about some words that you need to know as I'm going to go forward and start talking more about enzymes. You need to know what a substrate is, and that's the molecule or molecules that the enzyme works with. You need to know what the active site is, and that's the part on that molecule where the action happens, so to speak, right? That's where the substrate fits in so it can do its job. And when it does fit in, that is something called the induced fit, right? So these are things we already talked about. This is what we left off with on Monday. So again, you can see here, the substrate is the molecule that the enzyme acts on. And your first word for attendance is to answer me this question. So if the substrate is the molecule that the enzyme works on, so to speak, where does the substrate fit into the enzyme? What is that spot called? That is the first word for attendance. Actually, technically, it's a 2-word answer. So any questions about this slide before we talk more about how enzymes work?
What was the question again?
Question is, hold on, sorry, somebody wants to join the call. Let me fix this very quickly. I'm sorry, let's do this. Which reminds me, of course, right now I'm preaching to the choir, but if you're trying to join a call, because this is Google Meets through WVSU, excuse me, there we go. Because it's a WVSU Google account, or through WVSU Google account, you must be logged in to your WVSU Google account to join these meetings. I did see somebody just now, I got the notification and saw that somebody was trying to join who was not using the WVSU account. Luckily, I saw it, and I've now let them in. But from now on, remember, guys, and I wish I could fix this, I can't. You need to be logged into your WVSU email account to be able to get to the video without, or excuse me, get to the live meeting without me letting you in. So again, the question for the first word of attendance is, right, a substrate, I'm saying the substrate is what an enzyme acts on, right? That's its molecule that it works on. I'm asking you, where does the substrate fit into the enzyme? What is that spot called? So the attendance word, first word for attendance, if you will, is actually 2 words. So the answer is 2 words. All right, so let's talk a little bit more about how these things work. So again, we can see here, before I go to this next slide, here we have the enzyme, we have the substrate that gets attached to it, and you can see how the shapes perfectly match, kind of like a lock and key. And it gets in there, and then you got the induced fit where the enzyme kind of like hugs around it, so to speak. And you can see here the substrate's still there, but it's changed colors. What that symbolizes in this picture is that the chemical reaction has happened. So here we have this one product, this green product, and now we have, excuse me, this one reactant, right? That's that green thing, that's the reactant. That green thing, that's the reactant. Now here we have these 2 products, because basically in that reaction, that specific reaction, and there's all types of different ones, that one took a big molecule and broke it down into 2 small molecules. And the products then, as you can see, now have released, right? The products go their own way, look at that. Now the enzyme looks just like it did at the beginning. It is ready to go, which brings us to this. After the product is released, the enzyme is now able to accept another substrate molecule. Another way of saying that is the enzyme is not used up in a reaction. That'll be very important for the exam. I'm not going to ask a question. There will be no question where it says, are enzymes used up in a reaction? That won't happen. What will happen is, I'm going to give you some chemical equation you've never seen before. So I'm going to make it up. I'm going to make some gibberish words up. And if you see something like, let me write it down for you. A plus B, this was the chemical reaction, plus C gives us C plus D. And I say, okay, which one of those is the enzymes? This is when you need to know this bullet point here. Because look, the only thing on both sides of the equation is that C. So if you see something on both sides of the equation, that is the enzyme. Because it didn't change, right? It's just in there. Now technically, well, I don't want to get into that too deep, but a lot of times when we're talking about chemical reactions and deeper biology, usually we know what the enzyme is, but they usually don't put it in, I shouldn't say usually, but very often they don't actually write it into the chemical equation. But there will be a test question where I say, I give you some chemical equation and I ask you which one is the enzyme. So there you go. There's your first hint of how to know which part is the enzyme. If it's on both sides of the equations, it's the enzyme because it wasn't used up in the reaction. As we mentioned earlier in the previous week, when we finished up chapter 2, we were talking about proteins, right? And how they're very sensitive to their environment. And now, again, I'll remind you, as far as we're concerned, all enzymes are proteins. There are exceptions. You can look those up for independent work. But anyway, proteins are very sensitive to their environment. Enzymes are proteins, therefore, they are very sensitive to their environment. Here's some things you should know about enzymes specifically. Generally speaking, the warmer things are, the faster chemical reactions happen. Generally speaking. Now, granted, there is an optimum range. So if it gets too warm, it's not quite working as efficiently. Obviously, if it gets too cold, it's not working quite efficiently. And also, I'll tell you, every enzyme is different, right? Every specific enzyme works on a specific chemical reaction, and it has different optimal temperatures. As you might imagine, a lot of the enzymes in our body work very great at 98.6 degrees Fahrenheit. Of course, then remember, there's all kinds of other living organisms. So as you might imagine, these different enzymes across the world work at different optimal temperatures. So again, not only does it work not as optimally if it gets too hot, but then if it gets way too hot, we have something called denature. And that's something you should know. Denature basically means it melts it. So remember I showed you, we were talking about how the environment affects proteins in general last week. And I was talking about how those bonds, right? Bonds keep atoms together, right? When you talk about this molecule that has all these atoms, it's those chemical bonds that keep those atoms together. And it takes energy to do that. And heat is energy. So if you apply enough heat to these molecules, not just these molecules, pretty much any molecule, once you apply enough heat to it, you'll break the bonds. And in this case, that will just basically, again, denature, aka melt the enzyme. Also pH, we already talked about that, right? So if you have something, if this particular enzyme has this particular shape because there's hydrogen bonds holding it together. And remember, hydrogen bonds are this reaction between this partially negative oxygen and this partially positive hydrogen, right? That's what's keeping them together because they have that partial charge. But then if you talk about pH, if you had a bunch of acid, a bunch of H+, that's not partially charged, that's fully charged. So you can imagine that's going to disrupt those hydrogen bonds. Because that partially negative oxygen is going to be fully attracted to that fully positive hydrogen, right? Forget the partially positive hydrogen that it's been dealing with. Now it's a fully blown positive hydrogen. Same thing with base, right? So if you put a bunch of base in there, that's a full charge. So once again, yes, pH will change the shape and therefore the function of an enzyme. Also, salt concentration will affect how it works. You can read more about that on the exam or in the textbook. I'll just leave it at that. And then there's these things called coenzymes, and I'll also leave that at that because we'll talk about those later. So I would say I'm gonna put an X through the salt concentration because you almost definitely aren't going to need to know that for the exam. pH and heat, sure. I might ask some questions about pH and heat. All right. You know what? This is kind of out of order. I should put this bullet point right up here because kind of stop that conversation. So let me back up a step. Like I mentioned earlier, an enzyme is not used up in a reaction, right? So when I give you some fake equation you've never seen before, if it's on both sides of the equation, that's an enzyme. Because again, for the exam, I'm going to give you some equation you've not seen before and say, what's the enzyme? Which one's the enzyme? And I said, that's the hint, right? That it's on both sides of the equation. The other hint is, again, it should hopefully, or probably, almost definitely, you're not going to recognize this because I'm going to make some words up. So if the word ends with an ACE, then you're like, oh, that's the enzyme. The enzymes, as far as we're concerned, all end in an ACE. Like lactase is an enzyme, and I'll give you that example on the next slide, as a matter of fact. So again, it's not that you need to memorize all these different enzymes. You just need to be able to critically think. And if you see something that ends with ACE, and it's on both sides of the equation, right? So now you got two things to look for, or two hints, right? So they'll have both of those things. So hopefully that'll help you. Any questions about that side? Okay. And I'll mention this because I don't remember if I have it in writing in the upcoming slides. But not only will I give you some equation on the exam and say which one's the enzyme, I will then, right here, sorry. I will then also ask you, what is the substrate? So here's a chemical equation. Which one's the enzyme? Which one's the substrate? So the way you can figure out which one the substrate is, as far as we're concerned, all enzymes are named for the substrate. That's not true, but for the purposes of what you're studying and what you need to know for the exam, they're all named for their substrates for what they do. So for example, like I just said earlier, lactase. Lactase is an enzyme. So if you see it on this chemical equation, something that says lactase, and then somewhere else on one side of the equation or the other, because we're not talking about the enzyme anymore, we're talking about the substrate. If it says lactose, you're like, oh yeah, that must be the substrate, because the substrate is named after, the enzyme is named after the substrate. So that's how you figure it out. Any questions about this slide before I give you a visualization, again, of an enzyme doing its thing? All right, here we go. So there we go. There's your enzyme, right? In this particular case, it happens to be lactase, which is not a made-up enzyme. It's a very famous one, actually. Almost every biology student knows about it because it's used in so many examples. Anyway, there's the enzyme lactase, and it's ready for its substrate. Any guesses to what its substrate might be, given the fact that lactase and every other enzyme, as far as we're concerned, is named after its substrate? What's the substrate? And I just said it like 45 seconds ago. Any guesses? Substrate. Lactose. Lactose is the substrate. So there it is. Lactose is a disaccharide. Not that you need to know this. This is just an example. But remember, a disaccharide is two monosaccharides put together. The purpose of this enzyme is to break it down into its two parts. So the substrate comes into the active site, right? That's the substrate binding, aka the induced fit, right? Then it just so happens that since we're breaking this up, this happens to be a hydrolysis reaction. Therefore, in this case, we're going to need a molecule of water, as you can see right there. Then what we can't see right now is we have all the ingredients, right? We've got the enzyme, we have the substrate, we have the water, even though you can't see it here. You see there that it was added to it. Now it's ready to do the chemical reaction. Chemical reaction happens, the products leave. In this particular example, the products happen to be glucose and galactose. Now that enzyme is free to do its job again. Now I'll remind you once again, this is just an example. You don't need to know lactase. You don't need to know glucose or galactose or H2O, because again, they're not all hydrolysis reactions. All you need to know is that an enzyme takes in its substrate into the active site. Of course, that wasn't, into the active site. It does its job, the products leave, and now the enzyme's ready to go again. So again, as far as the exam is concerned, that's important because if it's on both sides of the equation, that if there's only going to be one thing that's on both sides of the equation, then that will always be the enzyme. Because the enzyme is not used up in a chemical reaction. It's making it happen. Anyway, any questions about that slide? All right, so that's how enzymes basically work. Let's talk about how there's some exceptions to it, and how things can be tweaked. Enzyme inhibitors. You need to know what they are. Enzyme inhibitors, well, it's just like what they say. They inhibit metabolic reactions. So if you were not in a biology class and someone said, what is an inhibitor or what is an inhibition? How would you answer that? Like if someone said, man, I went out with my friends at the Super Bowl, I went to a Super Bowl party and I lost a lot of my inhibitions. What do you think they mean by that? They lose their keys?
No, they lost their helping their team or something.
Or what? I think
They're helping their team, like.
Oh, well, I like that. Maybe I use a different example. Yeah, so they did that. They went to the Super Bowl party. Everything was fine. Next week, they could just go out to a bar with their buddies. They're drinking a lot, and he says again, man, I was out drinking with my buddies, and we all lost a lot. We lost our inhibitions that night.
They got drunk.
Yeah, and which caused them to do what? I thought maybe more people knew this term. Maybe I just drank too much in the Marines and heard too many people getting chewed out and losing their inhibitions. So an inhibition is something that keeps you from doing something, right? So when you go out and say you lost your inhibitions, that's like saying you probably kind of acted a little while because that inner voice that said, oh, you better not do that. That's an inhibition, right? It stops you from doing something. So same thing with enzyme inhibitors. They stop enzymes from doing whatever they do. Remember, each enzyme does something specific. So those specific inhibitors stop it from doing that thing. How do they work? You need to know this too. They work by binding to the enzyme. That's basically it. Of course, obviously, I'm going to get a little bit more detail, but for now, that's it. Enzyme inhibitors stop enzymes from doing what they're supposed to do, and they do it by binding to the enzyme. And that binding, when it binds to the enzyme, that's what disrupts the function. And I'll explain that later. Before I even get into that, because I don't remember if I have this in writing, I want to explain why this works, why we even need enzyme inhibitors. I mean, there's a lot of reasons, but here's one of the big reasons. So do you guys remember, I don't know if you're old enough, do you guys remember fidget spinners? They were like really big for like 2 or 3 years or something, huge. So imagine you're a toy factory, you make a variety of toys, but then these things start blowing up, and you're like, all right, we need to put all of our resources and employees on building these fidget spinners, right? Let's quit making other stuff for a while. We need to start making some enzyme or some fidget spinners because that's what's making our money. Then all of a sudden they're not as popular anymore. So now you've got a ton of fidget spinners. You, as the factory director, need to quit making fidget spinners, right? Now you need to start allocating resources and employees back to the other stuff, right? You don't need to make any more any more fidget spinners because it's a waste of resources. Same thing here. Your cells, not just your cells, any cell needs to make things, right? We need stuff. Cells need stuff, but they don't always need it. So very often what will happen is once they've made enough of that product, very often that product itself will act as the inhibitor. So then the enzyme's like, all right, I'm gonna quit building this product. So then the cell can use resources where they're needed, because they're no longer needed there. That being said, all right, so again, back to enzyme inhibitors, there's two types. The first type we're gonna talk about is called competitive inhibition. And you, by the way, you need to know the difference between the two types. And they're in no particular order, but this first one we're talking about is competitive inhibition. This is when the inhibitor, again, they all bind to the enzyme, right, regardless of which type. This particular one, where it binds to the enzyme, is the active site, which is very straightforward, right? So hopefully by now we've been using this word so much, and you've seen enough pictures where you can picture an enzyme with that hole, that particular shaped hole in it, because that's where the substrate comes in and that's where the magic happens, right? So a competitive inhibition, that's the process. So the thing, the competitive inhibitor plugs the site, like you can see in this meme, and that keeps the substrate from, attaching to the active site, to the enzyme, and therefore the enzyme can't do its job. And I like, even though this is just a meme that I found, I do like how, notice that the substrate is not identical, the inhibitor is not identical to the substrate. Sometimes it's very close, but in this case, it didn't even need to block both active sites. As long as it can just block one of the two, then hey, it's done its job. It's a good time to point out too, just for your own knowledge, a lot of pictures when you see like in textbooks and things like that when talking about enzymes, it's almost always like two little holes in the active site. It's not always like that, right? Think about all the different molecules you yourself drew in chapter 2. In the chemistry lab, right? So there's all kinds of different enzymes, but someone decided a long time ago that when we draw enzymes to teach people, it should be a circular shape with two little holes in it. So just so you know, it's not always like that. But anyway, that's competitive inhibition. Any questions about that one before I teach you about the other one? All right, just to see if anybody can guess this, and don't feel bad if you can't. I don't expect anyone to know it, but some people are just, I don't know if they're good at guessing things like this. If this one's called competitive inhibition, and the next one is also an inhibitor, what do you think the next one might be called? Nope, it's okay. The next one is called non-competitive inhibition. So here, the inhibitor still binds to the enzyme because, again, like I said previously, no matter what kind of inhibition we're talking about, that's how it works. The inhibitor binds to the enzyme. In this case, in non-competitive inhibition, the inhibitor doesn't bind to the active site because it's not competing with the substrate. Instead, it binds to some other spot on the enzyme. And by doing that, it changes the enzyme shape. And as I've said so many times, even when we're talking about proteins in general, but very specifically, especially when we're talking about enzymes, it's all about the shape. If you change the shape of that enzyme, you change its function. In this case, you stop its function, or at least maybe slow it down. There's another word for this. I'm going to put a question mark because I'm not sure which one I want you to know. Maybe I'll use both at once. Maybe I'll say a non-competitive inhibition, aka allosteric inhibition, and then ask the question. I don't know yet. We'll see. But yes, you should know that. Non-competitive inhibition. It's easy when it's just non-competitive and competitive, right? Competitive, to help you remember it for the exam, think about it. The substrate is competing with the inhibitor because they have the similar shape. They're competing to get to the active site, right? That's competitive. This is non-competitive because it's not competing with the substrate. It's like, you do your thing. Go ahead and try to get into the active site. I'm just going to go over here and attach to the enzyme here and change its shape. So you keep trying all you want. I'm not competing with you, but it's not going to work for you. So here's a picture of what I'm talking about. So first of all, this top one is the one the first one we talked about. That is competitive inhibition, right? So, this is just it working properly. It's the active site, the substrate fits in the active site, it goes there, everything works as expected. Then we have this, right, which is not written in words, but this is from the old textbook, so they didn't use these words. But again, this is competitive inhibition, the first one we talked about, where that inhibitor is competing against the substrate for the active site. It just blocks it. And then finally, the one we just got finished talking about, allosteric inhibition or non-competitive inhibition. So again, this time the inhibitor is not competing with the substrate. It's like, do your own thing, but I'm gonna attach to the enzyme, and when I do, I'm gonna change the shape. In that case, it changed the shape of the active site. Now the substrate can't fit, and it has been inhibited. Whew. So any questions about this slide? Again, this is just a visualization of everything we've talked about since we started talking about inhibitions, whether it be competitive or non-competitive. The next word for attendance is going to be answer this question. And there's going to be one word. It's a one-word answer. There's two types of inhibitors, competitive and non-competitive. Which type of inhibitor is the one that goes directly into the active site? So that one word will be the next word for attendance, whether it be competitive or non-competitive. That's the next word for attendance. Which one blocks the active site? All right. So again, that's it for talking about inhibitions or inhibitors. Let's talk about some other stuff. It's kind of like the opposite. So this time I'm not talking about slowing down enzymes. Now we're kind of talking about helping them do their job. Because very often enzymes do require helper molecules. It can form temporary or permanent bonds. So very often, like the molecule, the enzyme itself, not enough. Sometimes it needs more. And these things have names. The first name is cofactor. Cofactors are inorganic ions. So like iron and magnesium, right? So you've got like hemoglobin in your red blood cells, right? So when you breathe in and the oxygen is coming in, it attaches to the red blood cells. And that's how, because there's this cofactor. You have this iron inside your blood cells. So that's just one example. Obviously, as usual, I'm not going to say, what are the two examples I gave you for cofactors, right? I mean, that might help you remember, but I'm not going to specifically ask you about the examples. Cofactors are inorganic ions. Coenzymes, on the other hand, are organic molecules. They're usually carbon and hydrogen based. I'm probably also not going to ask you that. So basically, again, and this is not exactly correct, but you might, for this 100 level class, maybe think of it as like cofactors. Those are like small little metals. Coenzymes, those are actually molecules. Either way, both of them participate in these reactions without being altered. Therefore, they also are recycled and used. I'm going to put it next to that too, only because I talked a lot about that with enzymes. I kept beating that dead horse, right? That bullet point is also true for enzymes, and you need to know that for enzymes. But cofactors and coenzymes, I'm just going to kind of brush over it on the exam, if anything at all. So yes, this is a true statement. Yes, it's very important when we're talking about enzymes as far as the exam is concerned. Yes, it's very important as far as how things work is concerned, but as far as the exam is concerned, that bullet point when we're talking about cofactors and coenzymes is not very important. Also this too, I'm going to put it next to this. Actually, I put a question mark. Probably not going to ask you this, but just so you know, that's basically largely what enzymes are, or excuse me, what the vitamins are. Like vitamin A, right? Vitamin C, all these vitamins, very often they serve as coenzymes. So that's why you need them, right? So when I say you need like vitamin C for your immune health, that's how it's very, well, maybe that's not a good example. Anyway, yeah, and you can look that up if you want for independent work. Which vitamins serve as the source of coenzymes? You can give me a list. If you want to, you can get even deeper. Like, all right, so here's the list of vitamins, and here's the name of the enzyme that they work with. And here's what those enzymes do, right, if you want, right? So again, it's a good reminder because someone asked me this last week, or maybe it was earlier this week. For independent work, there is no minimum or maximum length of paper. Turn in 2 sentences, turn in 20 pages. It doesn't matter. You just keep turning it in until you hit 100 points. I do that for a reason. Some people like to write long stuff. Some people like to write short stuff. So do what you like to do. Any questions about this slide? Oh man, this slide was out of order. All right, let's pretend like I didn't just talk about that. Hit the time travel button and put your brain 5 minutes ago. This is what I was talking about earlier. I said this earlier when we first started talking about inhibition. And and I said, I'm not sure if I have this on the slides or not, so I'm going to go ahead and tell you. But turns out I did have it on the slide, just way out of order. So what I was mentioning earlier when I was talking about the fidget spinner thing, like, you know, your cells may need a product, but once they have enough of it, then they need to stop using resources like energy and material to make those things, right? Your cells don't need to waste the energy and the material to make those things because they have enough. And all that stuff I was mentioning, that's called feedback inhibition. So, which is the use of a product to regulate its own further production. Any questions about that? That's all in the name, right? And just to clarify things, just so you know, as far as work, I don't know if I want to say this or not. So remember we talked about competitive inhibition and non-competitive inhibition. This is not the third type, so it's not like there's 3 different types of inhibition. What I'm saying is like there's inhibition in general. As far as we're concerned, probably most of it is feedback inhibition, right? So feedback inhibition again is once your cell, the product itself is used as the inhibitor. That being said, you know, in feedback inhibition, then you have the 2 different types. There's either competitive or non-competitive, just like we talked about earlier. So I just want to be very clear. Feedback inhibition is not the third type of inhibition. It's not like it's competitive, non-competitive, or feedback, right? It's not like that. Anyway, any questions about, and again, that's just an example. So there's nothing to memorize with that picture, but are there any questions about that slide? All right, as I mentioned, starting last week, I'm now doing the announcements and the reminders towards the middle of the lecture because any given time, when we start class, half the people aren't here. So here we go. As I mentioned, and so far only one person is here. This is so ironic. The first exam, you can retake it, but you have to email me to request it. If you want to know the details, watch the video for Monday. So far, only one person has emailed me and said, hey, can I retake the first exam? And ironically, that person didn't even, I guess, listen to the lecture on Monday. So it wasn't like, hey, I'm writing to request like you asked. They were just like, hey, can I retake the exam? Yes, like I said on Monday, you can retake the exam. So email me. If you want to retake it, email me. Once again, as a reminder, you have nothing to lose. You cannot get a lower grade. If somehow, when you take an untimed open book exam that you're allowed to take over and over again, if somehow you can still never get a higher grade, then we'll just keep your original higher grade. Again, to remind you what I said last week, from now on, like I said, this is the worst semester I've ever had with people doing the pre-labs properly. I blame myself. I'm the instructor, so the buck stops with me. I'm doing something wrong, obviously. No, it's not. It's a lot of people. So because of that, from now on, everyone can always come to lab. So if you haven't gotten an email from me or something from me this week that says, hey, I see you in lab, you're okay. Just come to lab. Speaking of which, again, I'm putting it in writing here. I'm saying it. I'm putting it in writing. The next pre-lab you do is, again, going to have some video questions, followed by pre-lab questions as usual, followed by 4 sets of hypotheses. So there you go, right? You've been made aware. Also, speaking of lab, finally, because someone sent me this question in an email. This is a very good question. What do you do if you missed a lab previously? Even though I mentioned this already, you're supposed to do it. You're supposed to get the data from your classmates and do it. And as I've said before, I realize that's hard because you weren't in lab. So meet me. One of your classmates did this. They met me. We went through the lab. I talked them through it. I gave them the data. And then they turned in the lab. And it looked very good. I think they may have understood it more than some of the people who were here. But yeah, so let me help you. And not only that, and I know I'm repeating myself again, but still no one's taking me up on this. Before you turn something in, just let me look it over really quickly. Even if it's not like really good feedback, like how can I improve this? Just say, hey, will I get any credit for this? And let me just look over the big stuff. Like, oh yeah, you'll get some credit for that. So let me help you. It'll take 5 minutes if you meet me online. Normally, what I'm going to start doing moving forward, and I can't do it today because my kid's sick, what I'd like to do moving forward is I'm sitting here in lab and you guys are doing your experiments. Before you submit it, just come up and show me. Like, hey, will I get any credit for this? Take me up on that. And again, like I keep saying, after I've upgraded it, you can always meet me online to get detailed feedback and a 5% boost to your grade. So any questions about labs? That's almost all entirely what this was about. labs or retaking the exam? All right. So here's the harder stuff. I don't know if I want to say harder. It kind of is harder. The meat and potatoes of this chapter, right? So we learned about energy and metabolism. You know about the different types of energy and how chemical energy works, right? Like however much energy it takes to build a molecule is how much energy is released when you break that molecule, all those important concepts. Now we're getting into the really important stuff for this chapter. There's something called cellular respiration, which is where we break down molecules. Usually we talk about glucose. But we break down all kinds of molecules that we eat to release energy, and that energy is what's used to keep us alive. So what we're about to talk about is cellular respiration. And just so you know, it is broken down into these 3 stages. And before I jump into it, I'm gonna let you know almost entirely this portion of the exam, and this is gonna be a huge portion of the exam, is me saying, in which stage does this happen? And your choices will be glycolysis, citric acid cycle, or oxidative phosphorylation, which I'm gonna give it a different name. It's called the electron transport chain. So that'll be it, right? So for a huge majority of this exam, I'm saying, in which stage does this happen? In which stage is this produced? In which stage is this used up? Which of these stages happens inside the mitochondria? Which of these stages happens outside of the mitochondria, right? So as I jump into this, keep in mind, almost entirely, that's what those questions are gonna be like from what I'm about to teach you. It's me explaining something and you telling me in which stage it happened. There are a few exceptions, and I'll obviously give you those exceptions as we get there. So here we go. The learning objectives. Obviously, there's no need to write this down, but the learning objectives of this section are, by the end of it, you should be able to explain how ATP is used by the cell as an energy source. You might not know what that is yet. That's okay, because I haven't taught you that yet. That's literally on the next slide. And you should be able to describe the overall results in terms of molecules produced of the breakdown of glucose by glycolysis. Or better yet, by the end of this, everything I'm about to teach you, well, not everything, most of the stuff I'm about to teach you, you should be able to recognize it as happening in glycolysis, one of the 3 stages of cellular respiration. So here we go. Again, before we even talk about this, and this is going to be a test question. Good grief, if you don't remember anything else from what I'm about to teach you, at least remember this. And this is how I recommend studying this. Everyone's different, but generally speaking, this is how I recommend studying this. Focus on the big picture, and then start adding details. And the biggest picture from this chapter, in this section anyway, is what are we after, right? What's the use of cellular respiration? What do we need? What are we trying to get? What are we trying to make? And this is the answer, ATP. So again, going back to what we talked about earlier about enzymes, right? We need enzymes to start all these chemical reactions. And enzymes, the way they do their job is by lowering the activation energy. So we need energy to start all these chemical reactions that are happening in our cells. Without those chemical reactions, we're dead, right? So this is how important this molecule is, ATP, right? This is the thing that provides that activation energy. So even if we had enzymes that are lowering the activation energy, doesn't matter if we don't have ATP, right? ATP is the energy. That'd be like if you had something like something you could attach to your car and it automatically increases the fuel capacity. It's like, or not the fuel efficiency. It's like, I used to get 20 miles to the gallon. I attach this thing, and now I get 100 miles to the gallon. But, and let's also assume in this weird hypothetical situation, the world ran out of gas, right? So, okay, cool. You can now use, you get better gas mileage, but we're out of gas, so it doesn't matter. Same thing here, right? So an enzyme might lower the activation energy, which is great, and we need it, but we still need something for the activation energy. So again, big picture. This is what we're after in cellular respiration. It is all about ATP. Remember that before you start studying how do we make the ATP, right? Because that's what we're about to talk about. So anyway, for now, let's talk about ATP itself. It's a very small and simple molecule. Not that I'm going to ask you that. It contains potential energy for cellular work, right? Specifically, remember when we talked about potential energy, we said there's different types. So specifically, chemical energy, right? So it's however much energy it takes to build this molecule is how much energy will be released when you break that molecule down. Here's definitely going to be a test question. This is the primary energy currency for cells. This is the molecule that cells directly get their energy from. Not glucose, because that will be an option. Granted, we need glucose to make ATP, but cells don't take glucose and say, oh, I'm going to use this for the activation energy. No, we need ATP to do that. Not the sun, because that will also be an option. Because I did tell you earlier, the sun is the ultimate source of energy, right? That's where it ultimately came from. But specifically, directly, this is what energy, this is what cells need for energy, ATP. You can almost think of it as a battery, a rechargeable battery, and I'll get into that later. It powers the majority of the energy requiring cellular reactions. I like that it says majority because there are some exceptions. You can look up those exceptions if you want for independent work. So which reactions don't use ATP? What do they use? And like I said, they're basically like rechargeable batteries. And this is what it looks like. Not that you need to know what it looks like, but that's what it looks like. And I will point this out before we move forward. It's kind of hard to see, but this thing has a little, that's negatively charged, negatively charged, negatively charged, negatively charged. So those are negative charges. What I haven't told you yet is that oxygen itself is a very negative atom. And also double bonds are more negative than single bonds. So basically what I'm saying here is all those little parts are very negative. And like we talked about earlier in this chapter, like I said, what's on the outside of an atom? Electrons. And what do electrons do? They repel each other. So it's hard to squish negative things together, right? A negative and a negative repel each other. Then again, sort of a positive and positive. The point being, not that you need to know the structure, but again, this whole thing with structure and function, those things are so negative, they're really hard to push together. Therefore, it takes a lot of energy to build that molecule. Therefore, and this is the point I'm getting at, there's a lot of energy released when you break that molecule. So as far as we're concerned, however much energy it takes to build a molecule is how much energy is released when you break it. All right. Yeah. So before we move forward, next word for attendance is to tell me what is the primary energy currency in cells. You could just use the abbreviation if you want. You don't have to write out all 2 words. All right, moving forward. Well, are there any questions on the slide, actually, before we move forward? I'm sorry. Okay, let's talk a little bit more about it. The structure of ATP. Again, it's the whole name is adenosine triphosphate, so it's kind of in the name. You have this adenosine base, sort of like, or not sort of like, exactly like the base from DNA that I briefly introduced you to earlier, right? There's A's and C's and T's and G's. This is an A. But it's not just it's not a nucleotide. I mean, not like the ones we learned about, because it's not just the base, but we also have these phosphate groups attached to it. 3 of them, hence the name triphosphate. And again, they're like little balls, little phosphate balls. There's 3 little phosphate balls. They're very negative. They don't want to be next to each other. So it takes a lot of energy to cram them together. And again, therefore, once you can detach them, once you break those bonds, you're also going to release all that energy that took them to put them together. So what I'm getting at here is that we start with ATP. That's the fully energized molecule. That's the, if you want to use the analogy I was using earlier, that's the rechargeable battery that's fully recharged. And then when you use it, the way you use it is you lose one of those phosphate groups. You break that bond right here, and that releases all that energy. So now, instead of having 3 phosphate groups, hence triphosphate, you now have 2 phosphate groups. It's diphosphate. So those are the 2 chemicals, or excuse me, 2 molecules you're going to need to be familiar with. The fully charged battery, which is ATP, and the depleted battery, which is ADP. Any questions about that slide? All right. And again, I want to remind you, you don't need to know the structure. Right? So if you're studying, don't think like, oh man, I need to memorize which one's the phosphate group, which one's the ribose, and which one's the adenine. You don't need to know that, right? This is just a picture. So you can look at it while I'm talking about it. Here's a more common picture, especially for 100 level classes. So there's your ATP on the left, your adenosine triphosphate, your 3 phosphate groups. When you use it, the way you use it is by getting rid of a phosphate group that releases the energy. And now you're left with that adenosine diphosphate. And of course, some phosphate, wherever that thing went off somewhere. But there you go. So that's the basics, right? Your fully charged battery is ATP. Your dead battery is ADP. Any questions so far? All right, as I mentioned previously, the way this works, so far I'm kind of just saying, if you lose that phosphate group, that releases the energy, and that is a true statement. But we get a little bit more specific. They don't just go anywhere, right? Generally speaking, the ATP energizes other molecules by actually transferring that phosphate group to them. So they're not just going off into space or disappearing, because as we know, you can't destroy or create matter, right? So they have to go somewhere, and just so you know, that's where they go. I'm gonna put it next to this because I'm not gonna ask you that. The important thing, maybe I should put a question mark instead. I don't think I'm just gonna ask you that. We shall see. But yes, definitely you need to know ATP works by getting rid of that phosphate group. Maybe I'll get specific enough to say it gets rid of it by transferring it to the other molecules that it's working on. That needs the energy. And the next few slides I'm going to show you are examples of this happening. You don't need to memorize these examples. They're not from your textbook. They're only on the PowerPoint, but that's okay because, again, these are just examples. You do not need to memorize them. Are there any questions about what's on this slide before I show you some pictures of some examples of ATP energizing other molecules by transferring phosphate groups? All right, here we have a muscle, right? A muscle cell. Specifically, we've zoomed in to the cell and we're looking at the muscle, the motor protein within it. And again, just an example, you don't have to memorize this. So obviously it's gonna take energy for those muscle proteins to work and move. Where do we get the energy? We get the energy from the ATP. Specifically, the ATP is going to transfer a phosphate group to that molecule, which leaves us with ADP, right? Full battery, dead battery. And again, after it's all done, you have the dead battery and that phosphate group that is no longer attached to it. Next slide, that's just another example. This one will be a little bit more important later in the semester, barely. We're not going to talk about how muscles work later in the semester, but we will talk about this. We will talk about how things are transported in and out of the cell and how sometimes that takes energy. And that energy comes from ATP, right? Again, ATP releases that phosphate group, which releases energy. More specifically, it doesn't just release it, it actually attaches it to the molecule that needs the energy. Again, we have our full battery on the left, ATP, and our dead battery on the right, ADP. One more example. Here we're building a molecule, some random hypothetical molecule. We want to attach on X to a Y. How do we do it? We need energy to do that. So ATP gives X the phosphate group, right? It releases its phosphate group, that releases energy. Now that has energy to get put together. So now in the products, we have our XY molecule. And of course, on the left-hand side, we have the full battery of ATP. And on the right-hand side, we have the dead battery of ADP. Any questions about that? All right. So this exam, unfortunately, is going to have a lot of cycles. This is the first cycle I'm about to teach you, and this is the easiest one. And for some reason, your textbook left it out. I left it in because it's so easy. This is hopefully to give you 3 points on the exam. The ATP cycle. So first of all, I'll let you know the cells spend ATP continuously. Like you use a lot of ATP. That's recycled when ADP and the phosphate come back together. And it takes energy to do that. Just so you know, there's about 10 million ATPs that are consumed and recycled each second in each working muscle cell. So if you want to look it up for independent work, how much, and that's, again, that's one cell. So if you want to look it up for independent work, how much ATP do you use each second in your entire body? How much ATP do you use? Anyway, I'm going to put an X through that because I'm not going to ask you that number. But yeah, so this cycle, very easy. You've got the full battery of ATP, right? You deplete the battery, which means you lose the phosphate tail. Therefore, you're left with ADP, which is the dead battery. How do we recharge it? Well, we put the phosphate group back on. And we do that with some energy source, whether it be food or light, whatever. In this case, in this chapter anyway, we'll focus on the food part of it. So we take that energy from breaking down food, we squish that phosphate back on it, and then there you go, we're back where we started. with a full battery of ATP. So it's the easiest cycle that you're gonna have. Full battery to dead battery to full battery to dead battery. That's the cycle, that's the ATP cycle. Here's another picture of it. So ATP, full battery. Then you deplete that battery because your cell needed energy to do work. Oh, then you're left with a dead battery of ATP. How do we get it full charged back up? Well, we use cellular respiration, which is what this chapter is about, to squish these things back together. And then you have the full battery of ATP again. And that is 8:50. I will see you guys here at 10 o'clock. Don't be late. I guess that's it.
What was the second, Dennis question?
I don't remember. And that's a good question. That's kind of the key, right? That's part of the challenge. Make sure you're paying attention to all of it. Of course, you guys are here, so if you miss one or two, it's just less extra credit, but still extra credit.
It's never happened before, but it like erased my email run from my phone.
Oh, that sucks.
Yeah. I remember two, but
Yeah, I don't plan them ahead, unfortunately. But like I said, good news is you're here, so you're getting the points regardless.
So you said the hell with us.
I'm so sorry. It's not just you. I mean, it's